And if I'd ask Brad and Neil to join us at the podium, at the panel, we will have a pretty extended time for questions. So we hope that the folks in the online will put their questions in the chat. And just as a reminder, if you could please put your name or say your name before your question and your affiliation. And we can get started with the tool in the corner.
All right. So great presentations. And this is the first of probably many questions I'll have. But this first one goes to Charles because it's top of mind. Great work with Jeannie and UCSF as a participant. I'm proud of that. Our other UCs are not, unless they're hiding in that list that are coming. Why isn't every cancer center part of this yet? What is the problem with scaling at this point?
We're just a private club, and we were very picky. The problem is that, and this has to do with the last point I was making. To participate in Genie, we don't have a pot of money and pay for all the work that each institution has to do to participate. It's very much sweat equity. Institutions have to have already invested heavily in the infrastructure that's required to deliver the data elements and so forth. So we have open calls. Institutions have applied. I can't tell you whether the other UCs have or not. I don't remember. And we're very open. And then we decide we have enough money to front at least some of the startup costs. But of course I'd want to go bigger. And I'll just sort of as a counter, some people who could apply do not. I mean, are really ready to join, and they'd rather not. And that's about sandbox stuff.
Yeah. Thanks.
Kojo, I'll take you from the right.
Yeah. Thanks for a very stimulating series of presentations. My question is for Charles. As far as AACR Genie goes, I've got two questions. Are your input for diverse populations based on what you're given from each of the participating institutions, or you have a particular strategy for actually enriching that population as one of your goals stipulated?
Okay, I'll take that one first. We don't enrich, we just take whatever comes. We believe that numbers will win in the end. So of the 120,000, there's something like 28,000 non-European descent by self-reported race that just comes in across the transom based on the relative percentages of such patients seen at the institutions that are the sort of founding members. But I'm to the point I was sort of making with a tool. For a sort of largely African-American population at say a historically black medical college and hospital, they don't have the infrastructure yet and we don't have the money to give them the infrastructure yet. So it's a societal issue right now, but it's certainly something we have to tackle.
Well, my second question is technical. Have you had a chance to evaluate what the discrepancies are between assumed somatic variants and actual somatic variants in systems that test both constitutional DNA and DNA from the tumors?
I'm not sure I understand what's behind the question, but MSK data is the best at doing that because we, as you know, invested very early in doing matching normal.
What I'm saying is the entire data set you have, not all of it comes from tumor normal.
Correct.
So have you had an opportunity to explore what the discrepancy rate is in the systems that do not do tumor normal?
Yeah, so I tried. It was on that pretty complicated slide with the violin plots that the institutions that do not do matching normal, the implication of the result was that the error rate is higher in blacks and Asians than whites. Of course, can't prove it, but the assumption is that's because reference data is largely white.
Thank you.
Otis, did you have a question over there?
Great session, thank you. So my question is also about diversity. there are three really important components. One is equity, just making sure that we provide access to these tools and across so everyone has access. Second is bias, and of course how models get trained. And the third is really the science and the biology that we can learn from studying diverse populations. So it's just kind of an open question to the panel. I think Charles probably spoke to it most, but I think this is such a fundamentally important aspect along these three axes of equity, bias, and biology that we can learn. That I would love to hear just some more about that and what your experiences are.
I'll take the first crack. I'd love to just hone in on the access piece because I think that's really critical. Before I joined Flatiron, I spent about 2 decades plus studying why patients do or don't take part in clinical trials. You know, why in fact are clinical trials not representative? And we spent a lot of time addressing the issue or attempting to address the issue based on behavioral models of decision making. That, you know, if you tackle the attitudes that patients have about clinical trials and improve their knowledge about clinical trials, that they're more likely to go on clinical trials. My thinking has really evolved over time, and I think the attitudes and the knowledge, while important because they help you be prepared for a discussion about clinical trials, they're totally trumped by whether you have access to a clinical trial or not. Whether you have the ability to participate in research, was it offered to you? And so, you know, I'm trying to address that now by enabling clinical sites wherever they are in the community setting to be research sites. And the more we make research possible or look like clinical care, and I think, Brad, you alluded to this as well, the better off we'll be in terms of enabling access for all patients wherever they receive their care, and in doing so, the patients, you know, upon which the evidence is based will be more representative by definition.
So one of the easiest ways to address it, in my view, although it's incredibly hard, obviously, is where you're including folks in the research we're doing. So in the health system work that we did prior to joining Verily, we work with many of the largest faith-based health systems in the country, and the IDNs. And at many of those, it's a core part of their thesis of why they exist. They're already doing research. Every time there's a question that comes out about this stuff, the leadership of Bon Secours Mercy Health calls us every time and says, how can we be a part of that and how can we drive it? So I think pulling, you know, when you look at Common Spirit and many of the big health systems in the country, they have huge populations. So I think the first place is to go to those institutions that can really allow diverse representation within their databases and with their approaches and the patients they recruit. And then the second is having a UX team, a user experience team, and others that are really looking at the nuanced differences based on different geographies, by different classes of, you know, socioeconomic status and other things. And so that's huge. I mean, so for instance, I referenced the L'Oreal project we're doing. Our success is 100% predicated on finding Hispanic women and African-American women that are willing to come into our database. Otherwise, our project will fail. And so you can imagine we have a big team that's their entire focus. So it'll be really interesting, and we're kicking it off next week is our first real kickoff for it, but it'll be really interesting to see the learnings of some of these where I think more and more now that it's top of mind, more and more people are requiring it of all the work that we do. And I think that's actually a frame shift that's only really happened in the last year for us.
I am just going to add on a different dimension to it. So I spoke a little bit about our risk classification system. And we did find, this was externally developed, it was not developed by us, it's a validated method. And we found that it undercalled risk in Black women compared to white and Hispanic women in our population. And it has everything to do with how that initial algorithm was trained, and not on, here we are implementing it in a much more diverse system of care. But it also gives opportunity to say, how could we do better in the development of that next risk model, and being able to simulate even with our patient population what an alternative risk model might look like. Likewise, when we implemented the guidelines, the new 2018 guidelines from the American College of Radiology for supplemental MRI in women with a personal history of breast cancer, we found that Black women were about 10 points lower in the recommendation compared to white women in that same population, in our population, and it was mostly driven by breast density. They had less dense breasts, which in theory you might say might make it unnecessary for them to get breast MRI because dense-breasted women have a lower sensitivity for mammograms and might need additional screening. But to just expose that disparity in recommendation even for a supplemental screening at the outset based on the guidelines or biology or access is right there. Thank you. Otis?
So there's Rob Wynn from Massey Cancer Center. Just a quick question. So as I've been thinking and being fascinated by this sort of integrative approach, it struck me that as we were talking about where things are happening, I understand Kaiser and the Geisingers and the NIH centers, that are designated, but in almost everywhere I've been, that's not where most of the action is happening. And so in the context of how do we actually integrate, for example, or involve the NCORP, thinking about it differently. I was just thinking about, I've not heard once the NCORP, which is an infrastructure that's been set up, of how we could retool that to sort of get them involved, because those are patients and those are our communities. Or how do we get the community health centers or the FQHCs, which again is another national infrastructure. I think that in some parts while we're thinking about the high tech, we're not so much thinking about the high touch. And I think for both of these to actually have the impact, we're going to have another innovative sort of models about how do we actually tweak the infrastructures that are already in place that can give you more people and hopefully more accurate data. The question I actually had was, that's going to be one about how do you sort of see, I'll start with that one. How do you see us involving them and how do you see your programs getting those programs involved? Because I kept thinking we do have all these national programs, but I don't really hear them in any of our discussion.
Well, I just wanted to thank you for that question, and I think that I'll maybe punt to Neil for a minute as well, but one of the things that I did want to highlight about the clinical trial that Neil talked about, a collaboration between Flatiron Foundation and Roche, was that it didn't require a huge amount of clinical trial infrastructure in order to be successful. And you're speaking about community cancer programs who are part of the clinical trial network. And it didn't, because of the nature of it being a diagnostic, so like here we're trying to talk about evaluation of integrated diagnostics and how we can do that more easily. I remember what you called it, the intentional data, right? The intentional data collection that needs to be on top of whatever you need to do for a clinical trial in these contexts was significantly lower of a burden than you would have for a therapeutic interventional study. The diagnostics studies actually present an amazing opportunity for just a minimum amount of additional data to be collected and evaluated in the context of an EMR compared to the still incredible rigor that we need to do for therapeutic interventional studies. And perhaps thinking of it also from that perspective of involving diagnostics in the community health systems and the cancer programs might be another way of, and those are many of your customers, Neil.
Yeah, thanks, Mia, and thanks for the comment. We view the future success of the sort of national clinical trial enterprise to be tightly dependent on or associated with the expansion of clinical trials into sites that historically have not participated in clinical trials. And so to that end, we've started by enabling 80 of the sites in the community that use OncoEMR, which is Flatiron's oncology-specific electronic health record. We've enabled those sites to transfer data directly from the EHR into study-specific EDCs, and they already participate in retrospective data flow. The sort of next layer that is of great interest would be, in fact, partnering with existing clinical trials consortia that have particular needs and characteristics that could feed to the generalizability of the results that come out. So I'd love to talk to you about the NCORE afterwards. Yeah.
The only thing I would add is I think that as we've seen large networks come together in the country, the U.S. Oncologies, the One Oncologies, Florida Cancer, there are big footprints today that do a lot of clinical trials, right? But largely they're all commercially driven, and they're doing a lot of sponsored trials. And so if we want to really look at how to navigate that, I think we have to look at incentive structures and other things where I think the NCORs are less attractive, because there are, and the beauty of some of these huge networks is they have single EHRs, they have single infrastructures for their research, they have research leaders at a national level overseeing, you know, how many millions of patients that are seen. So I do think part of this all comes down to why somebody chooses to do a commercially driven trial versus some of the things that NCORE and other folks might find pretty compelling. And I think that one is actually solvable in some interesting ways.
Thanks. Last question I have, and I promise, no more. But this one is mostly in the context of Jeannie. So I was thinking about the collection and the talk about ancestry and the ancestral markers. And I was struck by we're still talking about black versus white versus Asian. When I think of ancestral markers, knowing that race is, by definition, a social construct, I'm reminded of the study by Lisa Newman and these guys who looked at triple-negative breast cancer for blacks, knew that it was a worse outcome, but then they looked at the real ancestral markers about who was from Eastern Africa versus Western Africa, and found out that there was a difference. We're starting to see that in prostate cancer and other things. So when we're pushing out Jeannie and all of these other things, and we're talking about ancestry, are we really pushing ancestry, or are we still looking at black versus white? Because when I think of white, I also think of ancestry to different parts in that. So I'm just trying to get a sense of, when we're talking about the ancestral marker, how deep in the ancestral markers are we actually going when we're looking at your data? Yeah, and I promise I'll leave you guys alone from now on.
I guess that's, for me, the answer is we're going very deep with what you can learn about ancestry from these panels. So we don't have, you know, whole genome germline, we don't have SNP panels, but I'm blown away by the accuracy of what's possible with, if you have a sequencing panel of roughly 500 genes, there's lots of extra DNA that's sequenced that comes out, that gets filtered out by the algorithms. So I hear you loud and clear. The race, the reason I use the word race is because we had self-reported race as one of the baseline data elements that we could get at the beginning that was low-hanging fruit to grab. But I agree, that will not necessarily be the way we think about this going forward.
And we have a similar algorithm that we've developed at Foundation Medicine, and it's more based on those geographic ancestries that you're referring to than self-reported.
And thank you for your presentation, because I'm going to reconsider Jeannie, so we should talk.
Hi, this is Joe Leonard's MGH. A follow-up part to this question. I very much like the idea of an ancestral genetic marker, and you can actually delineate that. 23andMe has genetic ancestry models. You can delineate even fractions, right? Your 0.3% Northern European descent, etc. Those algorithms, however, are based on phenotypes as the ground truth. Now, I'm posed to the whole group, what is a ground truth for something that's genetic if the genetic is based on location rather than the phenotype? So while I fully agree that a genetic ancestry model would be amazing, I don't think we currently have a ground truth that we can validate that against. I don't know what
That being
others
said,
are
I think
thinking.
that from a pharma perspective, there is a push for more diverse populations in therapeutic interventional studies, and to be able to be a little bit more formal about that, even with a model as flawed as it is, has some advantages for improving the diversity in the clinical trial populations.
I might just add, I appreciate the challenge, but I actually think the human genetics community, not the cancer community, has done a much better job of getting broader representation of different parts of the world in terms of genome databases. The tumors haven't been sequenced at that stage. So it's a bit of a hedgy answer, but I take your point.
I think we have some questions from the chat as well.
I'm going to read a question from Tommy Wang from the Moore Foundation. He says, as we move towards more research embedded in care delivery, as well as increased local evidence generation, how do we think about the balance between what types of evidence is generated centrally and related to regulation national standards, and what type of evidence should be more locally or regionally generated and inform local healthcare practices?
I think that it's both, right? I think that what we're trying to develop, and I'll be curious, is the Flatiron approach as well, is to generate it in the clinical care in the community, and give that data back to those community practices so that they can use it to inform care without question. And there's sort of a spectrum here in how we think about it of what can be, what traditionally technology is A/B testing, where you can just check 2 different navigator pathways or whatever a health system wants to understand, because there's no, regulatory concern or approval concern required. And so we're working to build some of the infrastructure to be able to do that work within the clinical care delivery model. The hope is still that gets aggregated and looked at broadly, and then the more difficult questions that require regulatory-graded data are really looked at with a much more sophisticated and intentional infrastructure. And I think that for us, the goal is to be able to automate a lot of that in the background as well, to come in and intentionally understand what does it mean to get a PRO and other things that the distinction how they would use it locally versus nationally is sort of irrelevant. It's where you have to get in and do chart abstractions and the rest that it becomes, I think, more complex.
Yeah, I would just add the point that as we move from data curation and aggregation and summarization to decision support, this question of that surrounds benchmarking, whether it's by a physician in your practice, by your disease group, by your institution, your region, whatever the cohort is that you want to compare, it becomes possible on the back end because the analytics can then be built upon the platform of curated data. So once you have the data, sort of the parsing it becomes a lot easier in some ways.
I'm struggling. I love the question of evidence generation and validation of these tools centrally versus regionally, and I'm struck by the bias that we experienced at our own institution of when you take an algorithm that was developed in one context and apply it to a separate population, you find that it performs differently in that population. And as we talk about equity in AI, making sure that we have the data to support the development of those algorithms from the very, very beginning is going to be essential to decreasing the risk that when you go to deploy it in a regional environment that you're going to get a different answer in its performance. And yet our country, our world is an incredibly diverse place, and that is a very, very tall order for us to be able to achieve as a country and as a world, as the people in this room who are charged with developing this next generation of tools. We have a lot ahead of us that we're going to have to accomplish, and to a certain extent, it's going to have to remain a little bit regional in order for us to move the needle and start small and get some traction and proof of concept. But understand that just because you can prove it in one environment doesn't mean it will automatically translate into another.
So George Wiener from Iowa. Actually, to follow up on that, I mean, the examples you presented are fantastic. What else is going on around the world where they do have different populations? You just don't hear a lot about that, whether there are international efforts that parallel what you're doing.
Well, Genie includes 4 European sites, and we're adding a site in Asia. Okay. So far, there's been no example like I think the question was asking or that Mia shared in which something completely different was coming from that data set yet, but those are a much smaller fraction of the total data.
I mean, I think we know from genomics that the incidence of EGFR-driven lung cancer is higher in Asians, and the incidence of certain cancers is higher, based on risk factors in various countries. So you're going to see a diversity of genomics when you do bring together a multinational data set. But I think what Charles hasn't shared was how challenging it has been to include our non-U.S. colleagues in some of the regulatory and data privacy concerns. It's an entirely different regulatory framework outside the United States, and it has been a huge lesson learned for that process. I'll let him.
Yeah, we've been in Singapore and in Israel in the last year looking at some of the work that they've done, and the work's incredible, right? Singapore, Temasek is basically is investing incredible amounts in Singapore in how to build population-level data sets that I think we could only imagine. And similarly in Israel, some of the work that came out of COVID was remarkable in terms of their ability to use the health system and the data they have at scale to answer questions around, does a booster work, right? Before any of us were doing it anywhere else. I think the challenge is how do we all bring it together and make it relevant to the US and actually synthesize it. Something obviously we and many others are talking about, but it is an incredibly high order when you haven't even got it right in the US to then try to solve it more broadly, so.
Mia, your comment about EGFR and then the earlier discussion about admixture and genetic ancestry, it wasn't the work of Jeannie, but it was of Dr. Carrot Zhang, who's part of Jeannie now. There's EGFR mutations in Chile, in Chilean women, I believe, are much higher, or approach Asia. And they weren't known to be Asian, but they have Asian admixture. And that has to do with the migration patterns from, however far back across the Bering Strait. So it's amazing what is there to be learned if we just collect the data and we do it responsibly.
Slightly different question.
Introduce yourself quickly.
Mark Stewart, Friends of Cancer Research. We've had our own experiences in terms of trying to look at potential variability across different kind of diagnostic assays that claim to have the same intended use. I think tumor mutational burdens, 1-HRD, and certainly other complex biomarkers. I guess my question is, and Dr. Sawyers, you kind of referenced this idea of harmonization of I guess, the genomic data that's coming to you, but I'm just curious, and for others, kind of what the extent of that is in terms of understanding or at least acknowledging the performance of different assays are likely going to be a variable and how that impacts the interpretability of the data that you're bringing together from different sites.
Yeah, it's a great question. It's bringing me back to the initial meetings that Mia and I attended. So basically it was if your test had not been LDT approved, meaning analyte reliable but not predictive value, we didn't allow it. We had to set the bar somewhere. But it's a whole different game now, and so maybe we should rethink that.
I would say the Genie data set has many diverse, for almost as many institutions as it has as many assays that go into the data set. And they're of different sizes, and so sometimes when there's overlap, 100% overlap in the assays for the coverage of a particular gene, you can ask those questions. But sometimes if there's, an assay that didn't cover a particular gene, the absence of proof isn't the proof of absence in that situation. And so it will limit your ability to continue to ask questions of that. But I might follow on from a theme you're talking about, which is the diversity in the testing platforms. I believe it was Nick earlier from Epic who mentioned ontologies and integration. And one of the things that I've noticed is that the testing menus across institutions are still very diverse. From an integrated diagnostics perspective and an evidence generation perspective, as we think about integrating all of this data together, whether it's going to be used for evidence generation or for clinical decision support, the fact that we don't even call the tests the same thing, a right-sided mammogram, a right-sided diagnostic mammogram, right-sided mammo, R-mammo, it's going to be different in every single institution. And even just to that level of being able to get an ontology for diagnostics, procedure type could actually go a very long way in all of us being able to leverage our data, not only for discovery and evidence generation, but hopefully in the future for complicated clinical decision support systems that want to come together and make insights from those. But so long as we still keep calling everything different names, your one instance of Epic or your EMR, you'll all call it something different. So just a plug for interoperability. Oh my goodness, the time is gone. Thank you so much for your participation in the room and online in the panel discussion. Thank you for your presentations, and I believe we have a short break, and we will resume. Thank you.